So,
Perfect.
hi, and welcome to the 13th TIA Centre seminar of the academic year. My name is Adam Shepherd, and I'm a postdoc here at the TIA Centre. For those that are new to our seminars, we invite researchers and leaders in computational pathology and associated areas to discuss their work. And today it is my pleasure to introduce Anurag Vaidya. Anurag is a fourth-year PhD student in the Health Sciences Technology program at Harvard and MIT, where he is supervised by Dr. Faisal Mahmood from Harvard Med School. Anurag completed his undergraduate studies in biomedical engineering and computer science at Bucknell University. His doctoral work focuses on developing tools that integrate multimodal clinical data in supervised and unsupervised ways to improve cancer diagnosis, prognosis, and biomarker discovery. And the title of Anurag's talk today is Threads, a molecular-driven foundation model for oncologic pathology. And thank you very much, Anurag, for joining us. We're really excited to hear more about your work. And when you're ready, please feel free to start.
Great. Thank you for the introduction, Adam. And I'm very happy to present here today. And without further ado, I'll get started. Um, so yeah, Adam mentioned this earlier, but a quick intro to myself. I completed my Bachelor's of Science in Biomedical Engineering and Computer Science from Bucknell University. And now I'm currently a PhD candidate in my fourth year. And I work at the Mahmood Lab at Harvard Medical School. So just to bring everyone on the same page, when we say computational pathology, what do we really mean by that? And at the center of the field, we have these digitized whole slide images of tissues, mainly cancer, but also very quickly expanding into non-cancerous and infectious diseases. But focusing on cancerous whole slide images, what can we do with these slides? Once these slides are digitized, they enable us to do quite a lot of interesting things. And I like to bucket them into two categories. The first is they can help us do clinical tasks better, such as predicting some clinical outcome or detection and segmentation of regions of interest, as just two examples. But they can also allow us to do some novel clinical tasks, which we rely on different tests for. For example, we can predict alterations to the molecular profile, or we can predict the prognosis of patients, which is not currently done in a very quantitative way and rather a subjective prediction. So what are whole slide images? Here I have a beautiful video of a whole slide image and we're scanning into it. And we see that we go from tissue phenotypes to more cellular organization to more cellular features. And all of this is to say that whole slide images are very large and they're very hierarchical in nature. They have important features at all different magnifications. And hence, it's not trivial to develop computational pipelines to analyze them or do any sort of deep learning on them. And the field has largely settled on a two-stage process called multiple instance learning to perform deep learning with these humongous slides. I'll refer to multiple instance learning as MIL for the rest of this talk. So to quickly go over MIL, there's first the pre-processing stage where we remove the background from the tissue to avoid any spurious correlations. And then we patch the large tissue into smaller tiles, which can be of various sizes, for example, 256 pixels. The first stage of MIL is really using a model to compress this patches into smaller dimensions. And then the second stage is to take these individual tiles and to make a slide encoding, which can then be classified for any task, such as grading, subtyping, or mutation prediction. Stage one is largely pre-trained and frozen because even with this two-stage process, it is not possible with current compute to backpropagate the signal to both the stages. Hence, we pre-train and freeze stage one and largely train stage two. So what solutions exist for stage two? The Mahmood Lab a couple of years ago published the CLAM paper in Nature Biomedical Engineering, and this largely proposed a framework to score, or rather provide a score for each tile in the whole slide image, and then aggregate these tiles with the scores to make a classification. And this was able to make some really cool heat maps. However, this is an expensive process, and it is really time for us to look at the next steps here now. So let's think about once we have our stage 2 largely set, what can we do about stage 1? In the CLAM paper and for a long time, we have been using the ResNet encoder, which has been trained on natural images such as ImageNet, as our stage 1 encoder. And in some ways, this made sense because it was trying to capture some very general image features. But in some other ways, it also doesn't make sense because it's not niche enough. It's not specific enough to histology images. So can we do better than that? This is where we recently also introduced the UNI slide encoder, which we trained on 100 million image patches from 100,000 slides, which were largely collected at the Brigham and Women's Hospital here in Boston. We trained UNI with the Dino V2 SSL paradigm using A100 GPUs. We also applied a lot of modern tricks to improve the diversity of data that we trained on, and you can find a lot of these details in the paper. And we also published one of the largest public evaluations for an image encoder. With this, we were able to show that this patch encoder that we developed can largely outperform the ResNet 50 ImageNet-based encoder, which is shown in yellow here. And this really shows that developing niche histology image-specific encoders is really the next step. So let's go back to our two-stage pipeline that we started this discussion with. Now, for stage 1, we have a strong patch encoder. For stage 2, we have the CLAM model and a lot of new upcoming schemes to do the classification. But there are some inherent challenges with the current way that stage 2 is trained. Training of stage 2 requires a lot of annotated data. For example, one of the tasks, which is largely considered to be a very stable task in computational pathology, is the PANDA challenge, in which you grade the prostate biopsies. However, there are 10,000 samples in this data set, and it requires a very large amount of data to get stable performance. This data is not only very costly to get, but it's also very time-consuming, and it also is prone to errors. We have often found that pathologists can make mistakes in labeling the slides. So with this expensive data, time-consuming data, and prone to error data, how can we really train our stage 2 model with a large amount of confidence in it? So this is where I would really like to shift our gears towards what I'm going to refer to as slide representation learning. And slide representation learning is really trying to focus on making stage 2 general purpose. So how can we train stage 2 in a task-agnostic manner and then have very little data to tune this stage 2 model to a specific task? So in very layman terms, this is taking the approach of something like UNI, but increasing its image dimensions from 224 pixels by 224 pixels to the entire humongous whole slide image. And what this will allow us to do is really operate on datasets which have inherently very, very small examples. For example, case stratification and prognosis predictions, these tasks, because it's very hard to collect data on outcomes, inherently there are going to be very small datasets. So let's talk about how can we make stage 2 general purpose. Let's take a step back and see what approaches do we have in our hands. First approach is a unimodal approach. What you can do is you can take the whole slide image, have an encoder model that will represent this image in a smaller dimension, and then have a decoder model that will try to reconstruct this image. Here, the assumption is that all of the information that you need to encode the whole slide image is present in the whole slide image itself. The benefits of this approach is that you can scale this to a very large number of slides. However, the limitation is that it does not leverage any of the complementary information that you have available for this patient. Another approach is a multimodal approach. Here, you can take the histology image, you can take genomic samples, you can also take IHC samples. The assumption here is that information to represent a whole slide image goes beyond itself. The benefits is that you can encode very rich complementary information. And in a way, my hypothesis is that it also provides some sort of regularization and more stable training. However, the limitation is that it requires multiple modalities per patient, which are not easy to acquire. And even if you are to acquire them, inherently, the clinical process doesn't allow you to have multiple modalities per patient. And this will lead to a problem of a lot of missing data. However, for the rest of this talk, I'm going to focus on the multimodal approach. So multimodal slide representation learning, what we are proposing to do is use the RNA expression of the tumor sample along with the morphology to guide the representation of the slide representations. And why do we really believe that this can work? Is expression data can offer a holistic view of the transcriptome. A lot of the phenotype and genotype interactions that are unknown to humans can be learned by comparing the morphology with the RNA expression. However, the core principle behind this is that it is free from any human biases and concepts such as grading. We have created these concepts to bucket patients by what we think is important. However, the transcriptome and the genome are the true underlying representations. So why don't we try and use those to guide our representations? To validate this approach, we published a paper in CVPR called Tangle, in which, in a very simple manner, we had a patch embedding SSL module. So this is your traditional stage one encode your patches into smaller dimension. And then you have a gene expression module, which was taking the gene expression data from these same tumor samples, compressing them to a smaller dimension. And then we would take the slide expression, the slide representation, and the gene expression modules, and just apply a simple info NCE loss, which is taken from the CLIP paper. To validate this approach, we really did two large experiments, in which the first type of experiment was to make sure the data efficiency of representations, in which we compared our model, compared our representations with an end-to-end MIL. And the other is the quality of representations per class. And what we did in this was called prototype classification. And you can think of this as a way to really test the quality of your representations in a non-parametric manner. Because what you're doing is you're taking your representations, you're making an average of them per class and defining your prototypes. And then at classification time, you're only classifying based on the distance to the prototypes. So there's no parameter involved in this. Some quick results to get you excited about this approach. On the left, what I'm showing you is two subtyping tasks. So breast subtyping and lung subtyping, in which you're classifying your whole slide images as either adenocarcinomas or squamous cell carcinomas for lung. And for breast, you're classifying them as ductal or lobular carcinomas. The size of the circles represents the training samples that were used. And we're comparing an ABMIL, which is a full end-to-end supervised approach, with an intra approach, in which rather than comparing your whole slide image representation with the genetic representation, you're comparing it with another whole slide image representation. So this is really a unimodal approach. And then Tangle, which is our multimodal approach. And you can really see the data efficiency of our approach here. With as few as one example, Tangle is able to outperform ABMIL with 25 examples. This is specifically about the breast subtyping task that I was referring to. On the right, we test what is called morphological retrieval, which is what I was referring to as prototyping in the previous slides. And here we are able to retrieve our samples at a much higher accuracy than any of the other approaches. So with Tangle, is the story complete? Uh, no, not really. And that if it was, we wouldn't be here really. So this is where I would like to introduce threads, which is going to be the rest of the focus of this talk. Um, so in threads, what we did was we really took the approach of Tangle, and we scaled it up to a very large data set. In this dataset, we really have four smaller datasets, which we collected from Brigham and Women's Hospital, Mass General Hospital, the GTEx consortia, and the TCGA. All of these datasets have their own interesting challenges and perspectives that they bring. For example, Brigham and Women's dataset is mainly a DNA dataset. So in this, rather than transcriptomics, we have DNA mutations and copy number variations along with paired histology images. Let's talk about GTEx a little bit. GTEx, on the other hand, is transcriptome, so it's all RNA expression, but it's all of the non-malignant samples. So these are not really healthy samples, but they're also not cancerous samples. So they bring a different perspective to the training paradigm. TCGA, as a lot of us know, is probably the largest cancer multimodal dataset out there. And then we also collected a niche MGH dataset where we have, again, transcriptome as well as paired histology images. So when you put all of them together, we have about 39 different organs. We have 47,000 plus whole slide images with a paired transcriptome or a DNA mutation assay. And again, these were from four different institutions. A very quick architecture overview. This is not a very complex architecture because we believe that simple architectures with really good data can help you scale well. So we have an RNA backbone, a whole slide image backbone, and a DNA backbone. And the purpose is to compress all of these modalities to a smaller dimension and then contrast them with the info-NC loss. One interesting addition here is that to compress RNA into a smaller representation, we use the SCGPT architecture, which is a foundation model that was made available by the Bo Wang lab last year, I believe. And the reason we do this is that data was trained on, that SCGPT model was trained on a lot of single cell data. So if we are able to take single cell foundation models and apply it to bulk transcriptomics, that is another interesting finding. So how do we evaluate threads? To evaluate threads, we have introduced the HistoQuest 54 dataset, which we are planning to increase to over 100 tasks now. So stay tuned for our updated paper. So in HistoQuest 54, we again have 54 tasks, which comprise of 26,000 plus whole slide images, and they have been collected from 20 plus institutions. To help the field go ahead with evaluations, we're open sourcing all of our tools. So as a part of this, there are three repositories which we have open sourced in the last 2 months. The first is PathoBench, which is Hugging Face for all of the splits. We plan to continue updating this repository with new tasks. We then have a PathoBench GitHub repository, which allows you to do large-scale validation of these models. And a very good use case of PathoBench is that if you want to evaluate 5 different models on 54 different tasks using 3 different image encoders, that can easily scale up to thousands of experiments happening simultaneously. We have done a lot of software engineering to make this parallelization of these evaluations very easy for the community. So please feel free to check it out and give us feedback. And then finally, we also introduced Trident, which is an image pre-processing pipeline, which is really supposed to be the CLAM 2.0 repository. So we open source all of these tools. Feel free to check them out and give us any feedback that you may have for us. Coming back to threads, for the rest of this talk, I want to share about 5 to 6 different insights that threads allows us to get. The first insight is that we're able to achieve the state-of-the-art performance on 50 plus tasks using threads and just a logistic regression model. So that we're only training a classification head rather than an end-to-end ABMIL or any other stage 2 model. We compared threads with three other slide encoders, which have recently been introduced to the field, namely Prism, GigaPath, and Chief. And all of them have their nuances. So Prism is encodes slides, but uses language as a guidance rather than the transcriptome. GigaPath is a unimodal approach in which they only use the whole slide image to encode the slide itself. And finally, Chief is similar to Threads in the sense that they both use an ABMIL model as their slide encoder architecture, but rather than transcriptome, Chief uses language. So it's similar to Prism in that sense. Taking a look at the full benchmark of 54 tasks, we're able to see that Threads is able to get almost a 6% gain over the next best model, which was Prism, and very comparable to Chief. When you break down the 54 tasks into some more finer categories, such as clinical subtyping and grading, mutation prediction, IHC status prediction, and most importantly, treatment response and survival prediction, you see that contrasting with the transcriptome gives a very clear benefit over contrasting with just language or contrasting within the whole slide image itself. And my hypothesis behind why this is happening is that language, while a very good modality, is only acting as a low-pass filter. It is taking all the knowledge that is out there about a cancer patient and it's refining it to what is clinically useful. So it is not able to provide the signal for something that is not known. Whereas transcriptome, because it's the ground truth, we're able to get much more richer contrasting signal.
Thanks, Anurag. Do you mind if I think we've got a question from the audience, Mustafa?
Perfect.
You've raised your hand.
Yes. Hi. Can you hear me?
Yes.
All right, good. Can you go a few slides back? Sorry. I was just yeah, this one. Yeah. Can you explain how do you handle the missing modalities in your data set? So some of the data sets, some of have transcriptomic data, some have DNA, right? So how do you do that?
Yes.
And
So we were very lucky that over here, we do not really encounter the problem of missing modalities. For each of the cohorts, we have paired histology with the transcriptome or the DNA data. So when you have a slide, we're always able to contrast it with something.
I see. Okay, so you have when you have DNA data, for example, you contrast it with DNA, and when you have RNA data, you contrast with that one. Is that
Yes,
correct?
that is
Okay,
correct.
cool.
And
All
in
right.
the later slides, I'm going to present another project called Madeline, where we do handle missing data. And just to give you an overview, the way we handle missing data over there was by using something called just contrast when you can. But I'll talk about that later if we end up having time.
Sure, yes. And the results section, sorry. So in
This slide?
the yes, this one, yeah, in Chief, what kind of language data did you use with Chief?
Yeah, so just to be clear, we did not train Prism, GigaPath, or Chief. We only
Uh-huh.
used their available weights. But to give you an overview of how Chief pre-trained their model, and please correct me if I'm wrong over here, whatever organ or disease the slide belongs to, they created a language representation of that and then
Mhm.
they contrasted their slide representation with this language representation. Whereas Prism, on the other hand, took the entire clinical report belonging to this whole slide image and compressed it and then contrasted it. Does that make
Yeah,
sense?
yeah, that's true. Yeah, yeah, yeah. But when you're using it, so for example, when you use Prism,
Yes.
um, did you use the clinical data?
No, we did
Then
not use the language arm of Prism or Chief. We only use the whole slide image arm.
I see. And is that the same with threads?
Yes, for threads, we do not, in this slide, we do not use the RNA or the DNA arms of threads. It's only the whole slide image arm.
Cool. Yeah, that's great. Thank you very much.
Of course. Any other questions, Adam, or should I
I don't think
continue?
there's any more questions at the moment. Thank you.
Okay. So moving on to the second insight that threads can give is that it's very data efficient and it's able to help you predict response to treatment. I say data efficient because you can see by the ends over here that there are always less than 100 samples for these data sets. You can imagine these are very small data sets. So over here, we are able to predict response to temozolomide treatment, which is a very common treatment for glioblastoma. We're able to predict response to bevacizumab, which is an ovarian cancer treatment. We have hormonal therapy for prostate cancer. We're able to assess neoadjuvant treatment in breast cancer and also predict survival in pancreatic ductal adenocarcinoma. While numbers are important, what doctors and clinical teams really care about is, are you able to stratify patients into different risk groups? Because that is how they are going to select their treatments. So in the bottom half of the figure, you can see that we've separated the patients into different risk groups, namely high and low, and Threads is able to get a statistically significant separation between these risk groups, whereas the other models, their risk stratification is not super clear. While I presented very small data sets, on the other hand, I want to present now very large data sets, but of rare brain cancers. This was really made available by the eBrains movement and institution, which collected around 3,000 samples of rare brain cancers and made that available to the public. On this, we give one sample, and then we ask the model which are the samples which are most similar to this test query, and then we see what is the accuracy on the retrieved samples. And you can see that just with one sample, 5 samples, and 10 samples, Threads is able to retrieve the samples more correctly. And why is this happening? If you take a look at the embedding space for eBrains, which is shown on the left here, Threads is able to just cluster these samples much better than any of the other slide representation models. Another insight that we gained from Threads is that multimodal models are robust to noise. So in this task, what we did was we found this cohort of slides which have been scanned on 5 different scanners. So same tissue, but scanned on different scanners. And in the bottom half, you see that we have made a plot of the hematoxylin and eosin stains present in these slides, and you can just see the large amount of variation present. Now, if you were to compress these slides using the slide encoders and cluster them by which scanner they were scanned from, if a model is robust to noise, it will lead to bad clustering, whereas if a model is not robust to noise, it will lead to good clustering by the scanner. When we ran this experiment, we see that Threads and Prism, both multimodal models, are not able to cluster the slides by the scanner, which is a sign that they are robust to noise. Whereas GigaPath, on the other hand, is able to do the clustering very well by the scanner. And this is really showing that multimodal signal, when training, can lead to good regularization and less effect by the noise. I also wanted to present one more quick result on the data efficiency side, and I think I skipped the order, so apologies about that. But I wanted to also mention we can further test the data efficiency of Threads by looking at something called as few-shot learning. And in few-shot learning, what you do is you reduce the number of training dataset size, number of samples in your training dataset, and then do the classification. So here, K represents how many samples you have per class. And because this is a 30 class problem, K equals to 1 really means a dataset size of 30. We can see that even with one sample, Threads is able to outperform most of the models, which are at even K equals to 8 or K equals to 4. And this really goes to show the data efficiency of Threads. We recognize that interpretability is very important to the field. And over here, we show that Threads is also an interpretable model. What I'm showing you over here is the diagnostic whole slide image. And on the other hand, the attention that is inherent to the Threads model. And this is to show that without giving it any specific signal to pick up on any specific morphologies, it's automatically able to look at the tumor for a squamous cell carcinoma slide. And then finally, I wanted to mention a novel capability that we have unlocked with Threads. And this is inspired by the language prompting, which is very common and popular these days. But now what we can do with Threads is molecular prompting. So you can create molecular prompts using any data set that you may have. And then you can ask Threads to find the whole slide image representation that is the closest to this molecular prompt. We tried this with multiple different data sets. And the most interesting finding for me personally was that it was able to do survival prediction much better with this molecular prompting paradigm. So the two examples here are TCGA kidney cancer, CCRCC, and TCGA pancreatic cancer, PDAC. And we're comparing it with what we call as mean pooling. So mean pooling is really taking all of the patches that you have for a slide and just averaging them to get the slide representation. You can see that giving a molecular prompt can be very powerful in predicting outcomes. As I mentioned before, I wanted to quickly touch on another project which is similar to Threads in the sense that it is slide representation learning, but rather than using the transcriptome or the DNA data as the guiding signal, we can also use different stains. So we have most commonly heard of the H&E stain, but in the clinic, it's very common to find other stains such as estrogen receptor and progesterone receptor and many others. So by contrasting these, you can also learn a very strong slide representation. We tested this on multiple different few-shot classification tasks, as well as large other tests, so feel free to check out our paper. And we again were able to show that slide representation learning can be much stronger than end-to-end ABMIL. So this really brings me to the end of my talk, and this slide represents a lot of the key contributions that I have been able to make, both in terms of stage 1 as well as stage 2. I've referenced other papers here. Feel free to check them out and email me with questions. But to really provide a quick summary of the talk, we have seen that H&E is available in the clinic, and we should use it to the best of our capabilities. And my real belief is that successful integration of AI and pathology is going to be the general purpose models that we're building, as well as going multimodal. And what's next from here is we really want to use these powerful models to focus to neoadjuvant therapy, or more recently, the antibody drug conjugates or immunotherapy. We would also really love to test these AI-powered biomarkers and assays in clinical trials And yeah, very excited to take the field ahead in this manner. I would really like to thank the entire Mahmood Lab in supporting all of these projects. And I'm happy to answer any other questions. And feel free to contact me at the provided email.
Perfect. Thank you very much, Anurag, for a really fantastic talk. That was really great. Sorry, our screen's just gone down here. Let me just load this up so everyone can see you again. Perfect. So, does anyone have any questions, either online or in person, that they'd like to ask Anurag? In that case, I'll start us off. So, this is really fantastic work. So, thank you for presenting. So, my question is, in terms of survival prediction models, it seems that the C indices that are generally getting are sort of around the 0.6 mark. So, not particularly strong in terms of survival prediction. Is it the case that foundation models are too broad, and that actually we need niche models for survival tasks, or what's your thoughts on that?
Yeah, that's a very good observation, and I have a couple of thoughts that I would love to share. First of all, I think C index is a metric that we have really invented to compare different models. And the C index, in my opinion, doesn't carry a lot of clinical implications. What carries a lot of importance in the clinic are the Kaplan-Meier curves and how can you stratify the patients into different risk groups. But I completely agree with you that C index of around 0.6 is not very robust. I think that general purpose models, more fine-tuned to the specific survival task, will be the way forward rather than a model that is trained from scratch for the survival prediction task. And we have seen this empirically quite often when we have taken, for example, we have taken threads and we have fine-tuned it rather than applying just a Cox regression model to the frozen model, that when you have the pre-trained weights in it, rather than training it from scratch, it performs better. But yeah, the C indices are always hovering between 0.6 and 0.75, which is not extremely robust.
Thank you, Anurag. And I'm not sure if I misunderstood something during the talk, but my question is, so obviously the framework in terms of the multimodal framework is to essentially regularize the training of the patch encoder, right? For the whole slide image essentially. So
Yes.
is it the case when you go to inference, you're just doing this based on the slide, or can you also use the DNA or the RNA information as well for inference? As I'm assuming that would also still give important information for the final prediction task.
Yes, so all the results I've presented have been only using the whole slide images, but you can very well use the pre-trained DNA and RNA encoders to do more tasks. We just haven't tested them out.
Okay, that's good to know. Thank you. And do we have any further questions from anyone in person or online I'd like to ask Anurag? Nasir's commented on the chat saying, "Great work, Anurag. Congratulations."
Thank you.
I've got a couple of questions if we have time. I think we have plenty of time,
Yeah,
don't we?
we've got a while, so please go ahead.
All right. So I start with a dumb question then. If you go back to Anurag, first of all, thank you very much for the great talk. I really enjoyed it. Great piece of work. So if you go back to the methods where you trained your model, so if I get it right, you have also trained the feature extractor in your method too, right? Or is it fixed?
The feature extractor is fixed. It's
I
the
see.
Conch 1.5 model.
I see. All right. So it's Conch. All right. And so uh so have you tried? So what I want to say is that so your method tries to train an aggregator
Exactly.
guided by the transcriptomic data or DNA data, right? But
Exactly.
what if we use, say, conch feature extractor data with like AppMill aggregator? Any of the algorithms that you've compared with, do they use the same feature extractor by but a different aggregator? What I'm implying is that how we make sure that this great performance is because of the aggregator or the feature extractor.
Yeah, that your question is on point and it makes a lot of sense. Um, the way that we have tried to address this question is by introducing a mean pool baseline in which we just take the patch encodings of the conch model and average them to get the slide representation, and then
Mhm.
compare the performance that you can get with the mean pool baseline with
Yeah,
our slide encoders.
yeah, it makes
I'm
sense.
not showing you results here, but feel free to check out our archive where we show that we're able to get almost an 8 to 9% gain over the mean pool baselines by the
Mhm.
slide encoding baselines.
Yeah, interesting. Yeah, I suppose yeah, that kind of answer that question. But obviously with not the most strong, not with the most strong or strongest baseline, obviously. But
Yes.
still it shows some difference. That's amazing. And also another question that I usually ask our great speakers at our seminars, that so feel free not to answer this, but it's not technical questions. What was the most challenging part of this project for you? And what would you do
Yeah.
differently if you're going to do it again?
Yeah, I'm glad that you asked that question because you never get to talk about the challenges. I would say data collection for this project was a very big challenge because we were coordinating between four different cohorts. I think as a field, we know a lot about language data, but we don't know a lot about how to do multimodal deep learning with transcriptomic data. So cleaning the data, pre-processing it, collecting it was a big challenge. And I think
Mhm.
the second biggest challenge was making the large evaluation benchmark um, and just being able to do the good software engineering bit to make sure the evaluations run smoothly. I would say those were the two biggest challenges that we had to overcome. And if I was to do this differently, um, I think I would really like to investigate more on the side of the RNA encoding arm. I
Mhm.
think
Mhm.
there's a lot of juice that can be extracted by playing around with specifically the encoder here. And if I had more time, I would love to have explored that more in detail from a more technical point of view.
Interesting. That was amazing. Thank you very much.
Of course,
Really,
thank you
really
for
enjoyed
attending.
your talk and great, great work again.
Thank you, Mr. Fair. So,
Can I ask a quick
yeah, of
question as
course.
well? So you mentioned the molecular prompting. In what form would you provide a molecular prompt? Like, how would that look like?
Yeah, so the molecular prompt is really you have say two different classes. You have RNA expression for both the classes. You take our thread's RNA arm, which is the scGPT model, which is a standard transformer model, and then you get the class prompts. Um, yeah, does that answer your question?
Um, so I'm not massively familiar with the that specific model. So in what form would you provide it? What sort of input does that model expect? Sorry, it's a silly
No,
question, but
definitely, definitely. So to break it down even more, what you would need is the name of the genes, which will be tokenized by the scGPT gene name tokenizer, and then you would need to give it the expression values, which are just it's just like a table. It's a row in a table, and those two things would be sufficient to get the encoding.
okay, yeah, that makes sense. Thank you.
Thank
Of
you.
course.
Yeah, I've got a question as well. So I also like the slide that you had in terms of interpretability in your heat maps. And I was wondering if you'd managed to speak to any clinicians or pathologists about interpretability in general and how you can provide more interpretability to your models.
Yeah. So there are two, I think there are two questions in there. The first is the threads interpretability, and then the second is how do you make models more interpretable? Did I get that correctly?
Yeah, that's right. Yeah.
Yeah. So with threads, our intention was not necessarily to make models more interpretable. It was just to show that you can use these models in the way that we are used to using traditional MIL models. Um, Without any guidance, you're able to get meaningful heat maps, which pathologists think for the task of subtyping, for example, make a lot of sense. So this is really showing that we are not taking away any capabilities that were present previously. In terms of how to make models more interpretable, which is another thing that I would have loved to explore with threads, going back to a previous question, is to inherently build the architecture of the model in a way that is interpretable to domain experts. And what I mean by that is making sure that the tokenization of your input of your modalities is meaningful. So, for example, instead of using patches as your input, you can use prototypes, which are just morphological patterns present in the slide. Instead of using individual genes, why can you not use biological pathways as your tokenization, which have very clear functional importance and relevance? So I think inherently interpretable models, which have tokenization that makes sense to domain experts, is one way of making your models more interpretable.
Perfect. Thank you very much, Anurag.
Of course.
Does anyone else have any questions they'd like to ask, either online or in person? If not, we'll draw the seminar to a close with a final question then. We normally like to ask our speakers what they think we're going to see ahead of the next 6 to 12 months in terms of developments in the field of computational pathology. Do you have any further thoughts or insights
Yeah,
on that?
I think I think something that I'm super excited to see upcoming in the upcoming year is how can we get rid of this entire two-stage pipeline and just have a single stage pipeline? I think with the all of the new paradigms around train short, test long that we are learning from NLP, I think if we are able to apply those to histology and just have one stage pipeline, that would be a very big advancement. And I'm looking forward for someone to do that.
Yeah, definitely. That sounds really exciting. But I think with that, then we'll call the seminar to a close. Anna, thank you so much for a really interesting seminar and for fantastically answering all the questions that you've had from us. And yeah, we wish you the best of luck with your future work and looking forward to see what else you do. And just a reminder for everyone else, our next centre meeting will be on Monday, the 14th of April, so next Monday, where we're joined online by Professor Peter Bohr from Aachen in Germany. And also a reminder that the Frontiers in Computational Pathology special session at MRUA, the full paper deadline is today at 11:59. So please make sure you've submitted if you haven't already, or that you do submit, sorry. But thank you very much, everyone online and everyone in person. And Anurag, thank you very much again for a fantastic seminar. And
Thank you
have a
for
good
the
rest.
invitation.
Bye, everyone.